we're here in San Diego for Bio. It's another great looking event. I didn't actually get down to the shell last night, but I did see the fireworks, which looked impressive. I don't know about you. Have you had fun?
Yes, it's been fantastic.
Right.
It's been really busy.
Oh, that's a good thing.
Yeah.
Could you introduce a little about your background?
Yeah, of course. So I'm Hayley Crowe, and I run our global life sciences business at Ecolab. Man, I'm a blue blood. I have been in the pharmaceutical industry my entire career. Yeah, so started off as a scientist, working in biochemistry and bioprocessing, and worked my way through at Pfizer and Novartis, and then found my way into figuring out how to provide solutions into scientists like myself to help solve those really tough challenges.
So what were you doing in the bioprocessing start early on? What were you working on?
Yeah, so back in the day when all of us did our own protein engineering and then scale up to actually develop our own assays, we had to do our own purification.
Yeah.
And so smaller scale, 5-liter scale, but I've lived through it. Back in the days when we all used E. coli.
Yeah, and I'm thinking gel electrophoresis as well.
Oh, we made our own gels. Oh, the days.
So now working at Purolite, I've been very interested in the jetting technology and some of the unique advantages it has. So could you talk a little about it? I'm going to ask a little about the sizes, but you start.
The jetting technology has really changed the way that we manufacture our protein A resins. And so historically, an emulsification process was used. And how that affects bead size, obviously, is you get this wide range of bead sizes. You get a pretty wide Gaussian distribution. Mm-hmm. So if you say something's 50 micron, it might be 25 to 105. Mm-hmm. And then there's a lot of post-processing that has to happen to sieve out the fines and kind of scale it down to the bead size you want. In jetting, you can actually dial it in. So instead of doing this kind of mixture and emulsified process, we actually use some acoustics and we push the resins right out. Yeah. We are able to dial the sizes in. And when we say 50, it's 45 to 55. And so you get a really nice tight distribution, but it also gives a lot of operational efficiencies in terms of how we manufacture. So less process steps, more consistent product in the end.
So I love the manufacturing side. So on the usability side, the mass transfer kinetics is improved, the flow rates are speeded up. What else can you talk about?
We're also finding that there's a lot of sustainability gains in it. So because you're getting higher yield as you're pulling through, you actually can run less cycles, and we end up reducing the total amount of water used as we go. And buffer usage as well. It's pretty incredible. Yeah.
Dynamic binding capacity, what have you got to say about that?
Yeah, so because of the way that our beads are formed, we actually can have a higher ligand density, and because of that we can get higher binding capacity. And our latest product that we just launched this week, yesterday, in fact, is proving to really deliver some of these higher DPCs.
Have you got some data on the website you want to point people to?
Yeah, we'll say up to 80, but I would say that it's very molecule dependent and process dependent, and so some customers are getting greater than that in their early testing phases.
Now, looking at this a little while back, I noticed that you had 45, 65 microns, 90 microns. Yep. Have you had customers asking for a mix of one size and another size?
Yeah.
And if you have, why would they bother to do that?
Not yet. I mean, I think this concept of being able to dial in the bead size is new to the market. I mean, jetting is new to the market. So to understand that we actually have the capability now to dial that in very tightly, I'm not sure anybody's really started to think about, well, what if we had a 50 mixed with an 80 together, and then what would the output be on that? And so, thinking through that a little bit, you could see potential applications where maybe you've got smaller beads that are maybe filtering out some of your smaller impurities mixed with maybe some larger beads that are actually capturing the antibody of interest. So I could see some applications where it could be useful. No one's done it yet, though.
Mm-hmm. Wow, you put the seed into somebody's brains.
Yeah, it might be something to think about.
So thinking about making that choice for which resin to use is not a lightweight choice to make because of the way the compliance and regulatory pathway, you really kind of have to stick with one of those choices early on, and that goes from scale up to mass production.
Commercial scale, yeah.
What do you tell people who ask you about the supply chain resilience for the new products?
Yeah, and some of the challenges. So one of the things that Ecolab has really invested into Purolite is ensuring that we are ready for commercial scale when we launch a program. So when we are shipping out samples at launch, they're actually coming off of our commercial scale validation batches, which means we're ready to be at the commercial scale from the beginning. So I think that's one big point in terms of thinking about when you're bringing in early new products, making sure that they're not just research grade. You got to ask the question, hey, is this ready for commercial scale? And if the answer is no, your next question should be, when will it be? So there's that bit. I think the other side to it is the security of supply. So, of course, we have our plant in Wales. We're opening a new plant in Landenberg so that we've got dual source supply with full lines and capacity in different regions so that you can actually get products should something go wrong. I mean, I think during COVID, all of us experienced this shortage of supply, of course. And while there isn't necessarily a shortage of supply today, I think the pain of living through that of what if that happened again and are we ready and are we prepared? It just made everybody look at this completely differently. And so we've made significant investment to make sure that if there was another, hopefully not COVID, that came across, we'd be ready to go.
Yeah, it's very reassuring. So in the couple of days I've been here talking to customers and friends, ADCs has come up a lot. So from your customer base, what are you hearing in the demand side or on the excitement side?
Yeah, so I would say the diversity of molecules has really, really, really increased. So what used to be maybe vanilla mAbs or just very, I would say, non-complex molecules has transitioned into this plethora of different molecules. And that actually makes a big change for us in terms of purification resins. It's not a one-size-fits-all anymore. And having a toolbox where you're able to test different protein A resins to give you the best yield, decrease aggregation, best operational efficiency, it's really important to test the different resins at this stage and not just stick with the same thing that always worked for the last 30, 40 years, because guess what? Molecules are different now, and they behave differently. So.
That's a good segue. So what about the future? What plans have you got for?
Well, we'll continue, obviously, to innovate in the resin space, for sure. I mean, there's a lot of opportunity, especially as the diversity comes out. I would say not just in protein A, but also in new modalities. We like to think about a lot of LNPs and exosomes and all these different things of the future. But I would also say in terms of polishing resins and downstream resins as well. So that's kind of what I would call stage one. But I think I would be remiss to not mention the big push into AI. So thinking about the future in terms of process efficiencies, looking at how we can optimize our processes, not just using algorithms and technology, but then also having Gen AI on top of it to help us be better as humans.
Yeah, no, it does seem to be starting to pay dividends now. There
It's
was a lot of hype, but now I think there are real...
Well, and I think the thing is there's a lot of fear as well of, okay, how do I make sure my data doesn't get out? How do I make sure my process doesn't get out? So you have to take this into consideration as you're thinking about it.
Yeah.
But I would also argue that if you don't start thinking now, everyone else will be. You will be left behind. So as I think about the future, I think about it's the companies that embrace it and figure out the parameters to live in that space that are going to be really, really successful. And if we let fear get in the way of what potential future could look like, then I think those are the companies that are probably going to struggle because not everybody's going to not do it.
Yeah, so not everybody's not going to do it. And the molecular diversity point you mentioned earlier puts more of a premium on that.
It does, really does.
Well, thank you, Hayley. It was a delight to talk to you.
You as well. Thank you very much.
Thank you. Cheers.